Human cells break down shared proteins more slowly than mouse cells, contributing to longer developmental timelines across species. This is Planetterrian Daily, episode 161. Researchers from the Ibisuya group at EMBL Barcelona and POL TU Dresden, along with the Savitsky team at EMBL Heidelberg, compared degradation across 4,000 shared proteins. Human proteins showed a consistent tendency towards slower breakdown than their mouse counterparts. This difference aligns with the observation that humans reach developmental milestones over longer periods than mice. The work points to protein stability as one molecular factor shaping species-specific timing. The comparison covered proteins present in both species and revealed a directional bias rather than random variation. Protein degradation rate therefore emerges as a measurable contributor to the pace of development. Protein degradation rates help explain why humans develop more slowly than mice. Across thousands of shared proteins, human versions break down more slowly, extending the time required for cells to complete developmental programs. This mechanism operates independently of differences in gene sequence and offers a measurable cellular clock. The finding opens questions about whether interventions that speed or slow protein turnover could influence aging trajectories later in life. The same principle may apply to other long-lived species where protein stability contributes to extended cellular maintenance. Most people assume developmental speed is set mainly by the number and timing of genes being switched on. In reality, the stability of the proteins those genes produce also matters. Right now, inside your cells, enzymes are continuously tagging proteins for destruction and replacing them with newly made copies. The pace of that cycle differs between species. Across 4,000 proteins examined in one study, human versions lasted longer before degradation than the same proteins in mice. That single difference stretches the window during which cells can grow, divide, and specialize. The effect appears consistent rather than limited to a few key proteins. The practical takeaway is to watch for future experiments that deliberately adjust protein half-life in model organisms to see whether development speed can be tuned without changing the underlying DNA. Now, shifting to a very different area of research. In mouse models, the Alzheimer's risk variant APOE4 raised levels of the protein NEL2. Elevated NEL2 caused neurons to shrink and increased activity in memory circuits. This early hyperactivity later correlated with poorer memory performance in the animals. Reducing NEL2 in adult mice reversed the structural and functional changes. The findings suggest a potential window for intervention before clinical symptoms appear. The study links a well-known genetic risk factor to a specific downstream protein that alters circuit behavior. Next up, work on human brain cells in culture. Peppercorn-sized clusters of human brain cells maintained in culture for a record 7 years followed aging patterns seen in living brains. The cells appeared to register elapsed time through changes that matched in vivo progression. Researchers noted this as evidence that intrinsic cellular programs track chronological age independently of the body. The model offers a platform for studying long-term brain aging outside an organism. No external systemic signals were required for the observed progression. The clumps retained structural and functional features consistent with normal brain maturation over the extended period. On a different note, comparative biology offers new angles on aging. Comparative analysis suggests overlapping traits between long-lived dog breeds and human centenarians. The work examines cellular and molecular patterns that appear conserved across the two species. Researchers highlight these parallels as a route to study aging mechanisms without relying solely on short-lived laboratory models. The approach could help identify factors that extend health span in both. The study draws on existing lifespan records for dog breeds and human demographic data. Ongoing studies aim to pinpoint which shared traits most strongly predict lifespan. From cross-species patterns, consider how temperature shapes traits during development. Experiments at the Smithsonian Tropical Research Institute exposed developing blue morpho butterflies to elevated temperatures projected under future warming. The wing scales responsible for iridescent color showed structural changes that lowered brightness. Both bird predators and conspecific butterflies would likely perceive the altered coloration. The result provides the first direct evidence that thermal conditions during development can modify this visual signal. No genetic changes were involved. The effect arose from altered scale architecture during growth. Turning to ecological recovery on former farmland. A former agricultural field left undisturbed developed into a diverse wildflower meadow within roughly 10 years. Plant species richness doubled, and thousands of orchids established without deliberate seeding. The outcome indicates that passive recovery can restore biodiversity at low cost when surrounding seed sources remain available. The site now serves as an example of natural regeneration potential on post-agricultural land. No active planting or soil amendment was applied after abandonment. Longer monitoring will track whether the orchid populations persist and expand. Researchers developed a method that reduces sintering time for three-dimensional printed zirconia crowns from up to 100 hours to less than 30 minutes. The shortened process maintains the mechanical strength required for permanent restorations. If scaled, the approach could allow same-day fabrication and placement of custom crowns. Current dental workflows often require multiple visits and external lab processing. The advance targets the thermal treatment step that previously dominated production time. Clinical testing will determine fit accuracy and long-term durability in patients. 2 ETH Zurich studies tracked brain activity in younger and older adults who drew together weekly over several weeks. Closeness between participants increased, yet similarity in their brain activity patterns did not rise in a straightforward manner. The findings challenge the assumption that interpersonal closeness necessarily produces more aligned neural responses. Age group differences in how coordination emerged were also observed. The protocol used repeated joint drawing sessions to measure social bonding alongside simultaneous brain recordings. Further experiments will explore which aspects of shared activity drive social bonding. A study of purchasing data showed that new limits on sugar-sweetened beverages in the Supplemental Nutrition Assistance Program coincided with reduced soda buying. The decline was described as meaningful yet modest by one of the study authors. The policy change targeted a specific category within a large federal nutrition program. Researchers tracked household-level shifts following implementation. The 12% reduction was measured against prior purchasing patterns in the same population. Additional analyses will assess whether the reduction persists and whether other food choices changed. Research from UNC Chapel Hill examined whether classroom teaching assistants represent an underused route into licensed teaching positions. The analysis found that many assistants already possess relevant experience and interest in classroom roles. Districts facing shortages could target this group with streamlined certification pathways. The study draws on workforce data to quantify the size of the existing pool. Teaching assistants often work directly with students and already meet some certification prerequisites. Pilot programs testing targeted support are now under discussion. Researchers decoded a cellular process that allows some cancer cells to evade immune detection. The work focuses on surface markers and internal signaling that collectively mask malignant cells. Laboratory models demonstrated that disrupting the identified pathway restored immune recognition. The discovery provides a new molecular target for therapies aimed at visible tumors. The mechanism involves coordinated changes in how cells present themselves to immune surveillance. Clinical translation steps will test safety and efficacy in patient samples. The China Manned Space Agency postponed a planned research mission to the moon after reviewing launch parameters. The delay follows a recent launch anomaly and occurs amid broader efforts to achieve crewed lunar landing by 2030. Mission hardware remains in preparation while conditions are reassessed. The agency provided no revised launch window in the initial statement. The unmanned mission forms part of a sequence intended to support later crewed landings. International observers will monitor subsequent scheduling updates. Before we go, keep an eye on further tests of whether adjusting protein turnover rates can alter developmental timing in additional model systems. That covers today's science and health news. Share this with someone who's curious about the latest research. I'm Patrick in Vancouver. See you next time. And if you'd rather watch than listen, find us on YouTube at NERA Network. Links in the show notes. This show comes to you from the NERA Network. If you want another sharp daily briefing, Unintended Consequences has you covered. True stories of good intentions that backfired. All our shows are free at neranetwork.com. Prefer reading? Today's full write-up, sources, and transcript are on the episode blog at naranetwork.com. Same briefing, readable format. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.